Welcome back to my new series of video session on the 6th chapter titled Molecular Basis of Inheritance. In this chapter, we will focus on the molecule of life or the blueprint of life, which is the DNA. We will discuss the structure of DNA, its process of replication, transcription and translation. An elementary concept of gene regulation, human genome project and DNA fingerprinting will also be discussed. Let me begin the chapter by explaining the series of classic experiments that led the search for the genetic material. And the learning goals are: 1. Explain how Griffith's experiments showed that a hereditary factor was involved in bacterial transformation. 2. Summarize how Avery, MacLeod and McCarty's experiments identified DNA as the transforming material in bacteria. And 3. Describe how Hershey and Chase's experiments led to the definitive conclusion that DNA is the genetic material. If we look at the timeline of the history of modern genetics, it began with the work of Gregor Mendel in the year 1865, where Mendel established factors as units of heredity. Later in the year 1869, a Swiss physician named Friedrich Miescher was able to isolate DNA for the first time from the nucleus of WBC cells. Miescher actually identified it as an acidic substance rich in phosphate, and he called it as nuclein. It was named as nuclein because he found these acidic substances in the nucleus. The term nuclein was later renamed as nucleic acid by Altman. Now we also know that a German anatomist named Walter Fleming was the first person to visualize chromosomes as thread-like colored bodies in the nucleus of the cells during mitosis. And he named chromosomes in the year 1882 as chromatin. Now in the year 1902, Certain and Boveri developed the chromosomal theory of inheritance. And this theory identified chromosomes as carriers of heredity. The experimental evidence for the chromosomal theory of inheritance came from the work of T.H. Morgan in the year 1910, where he demonstrated that genes are located on chromosomes in Drosophila. Scientists in the early 20th century became convinced that chromosomes were carriers of genes, or chromosomes were carriers of heredity. But they were also aware of the fact that chromosomes are made up of, or chromosome consisted of, both proteins and DNA. So the question was, which was the genetic material? During the time, scientists believed that since proteins are made up of 20 different amino acids, as opposed to DNA, which are made up of only 4 different nucleotides, so they believed that having more number of building blocks offered more variations and complexity to the proteins, and thereby many scientists considered proteins to be a preferred genetic material over DNA. Let's go through the classic experiments that led to the discovery of genetic material. The first headway was made by Frederick Griffith, a British bacteriologist, in the year 1928. And he studied 2 strains of Streptococcus pneumoniae, a bacterium that was responsible for causing pneumonia epidemic during the time. And Griffith was interested in developing a vaccine against pneumonia. Now the 2 strains that he worked on was rough strain or R strain and smooth strain or S strain. Both of these strains differed in their appearance. Now when R strain Streptococcus bacteria were cultured or grown, they formed rough colonies, whereas when the S strain Streptococcus bacteria were cultured or grown, they formed smooth colonies. And the smooth appearance of the S strain was due to the presence of a mucus capsule made up of a polysaccharide coat, whereas the rough appearance of the R strain was due to the lack of the mucus capsule. Griffith conducted a series of experiments in order to study the effect of Streptococcus pneumoniae on the mice. So in his first experiment, he injected R strain Streptococcus pneumoniae to the mouse. And what he noticed was, on injecting R strain, such mice did not become sick and they were all able to survive, or they lived. Whereas in his second experiment, he injected the mouse with S strain bacteria. And when S strain bacteria was injected to the mice, all of these these mice developed the sickness and eventually they died. So through his two of these experiments, Griffith was able to realize that our strain was the non-virulent strain or the non-pathogenic strain. So our strain was not able to cause pneumonia or the disease, whereas S strain was the virulent or pathogenic strain. So S strain was able to cause the disease. And he realized that the virulence of the S strain was due to the mucus capsule that was present in these bacteria, which protected the bacteria against the mammalian or the mouse's immune system. In his third experiment, Griffith used heat to kill the S strain bacteria, and he then injected heat-killed S strain bacteria to the mouse. What he noticed was mice injected with heat-killed S strain bacteria did not become sick and they all survived. They lived. So this shows that heating killed bacteria. But Griffith did not stop here. He wondered what would happen if he mixed heat-killed S strain bacteria with live R strain bacteria. And what he expected actually was that all of such mice injected with a mixture of heat-killed S strain and live R strain should survive because both of the heat-killed S strain and live R strain did not cause any harm when they were injected alone. But to his shocking surprise, when he injected the mixture of heat-killed S strain and live R strain to the mouse, all the mouse started developing pneumonia and eventually died. So what's going on here? When Griffith analyzed blood samples of such dead mice under the microscope, he found that there were live S strain bacteria in their blood. And when such S strain bacteria were allowed to reproduce, they even formed new colonies of S strain bacteria. So this thereby led Griffith to conclude that there was something in the heat-killed S strain bacteria that got transferred to the live R strain bacteria, making the live R strain bacteria to change or to transform into live S strain bacteria. Through his experiments, Griffith finally concluded that a chemical material from heat-killed S strain bacteria, which carried the genetic information to synthesize mucus capsule, had transformed live R strain into live S strain bacteria. And he called this phenomenon the transforming principle. Unfortunately, Griffith was unable to identify the exact biochemical nature of the transforming principle. Scientists around the world repeated Griffith's experiments and attempted several different ways to discover the transforming agent. Over 15 years later, in the year 1944, three American scientists, namely Avery, MacLeod and McCarty, succeeded in determining with the exact biochemical nature of transforming principle. They conducted biochemical assays through a series of elimination experiments using hydrolytic enzymes. Hydrolytic enzymes refers to a group of enzymes that are capable of breaking down chemical bonds in large macromolecules through hydrolysis. Since S strain bacteria was chemically constituted by carbohydrates, lipids, RNA, proteins and DNA, to determine which of the chemical constituent was responsible for transforming principle, they subjected heat-killed S strain bacteria separately with hydrolytic enzymes that specifically degraded or destroyed each chemical component. For instance, they used the following hydrolytic enzymes, like heat-killed S strain bacteria were added with carbohydrases. Now, on addition of carbohydrases, these enzymes would be degrading all of the carbohydrates in the S strain bacteria. Similarly, they use lipases to degrade lipids, RNases to degrade RNA, proteases which degrades all the proteins, and DNases that degrades DNA. So the basic theory behind these elimination experiments using hydrolytic enzymes was that elimination or destruction of a potential transforming material from the heat-killed S strain bacteria would prohibit transformation of R strain bacteria. As you can see in the illustration, Avery and his colleague initially subjected heat-killed estuarine bacteria with specific hydrolytic enzymes. Moving from right to left, in the first experiment, they treated heat-killed estuarine bacteria with carbohydrases to eliminate carbohydrates. In the second experiment, lipases were added to eliminate lipids. In the third experiment, RNases were added to eliminate RNA. In the fourth experiment, proteases were added to eliminate proteins. And in the fifth experiment, DNases were added to eliminate DNA. Such pre-treated heat-killed estuarine bacteria was then mixed with live R-strain bacteria and injected to separate mice. What they observed was, in experiments 1 through 4, where carbohydrates, lipids, RNA and proteins were eliminated, the mice still suffered pneumonia and eventually died. Additionally, they also recovered live estuarine bacteria in the blood samples of these dead mice. Contrastingly, in the experiment where they destroyed DNA of the heat-killed estuarine bacteria with DNases and co-injected with R-strain to the mice, all mice were healthy and they all lived. Moreover, no live estuarine was recovered from the blood of these living mice. These observations or results clearly demonstrated that carbohydrates, lipids, RNA and proteins were not the transforming material or principle, since transformation occurred even in the absence of each of these chemical components. However, on elimination of DNA, no transformation occurred in the live R-strain bacteria. Hence, it is evident that DNA was essential for transformation. Based on the results of their elimination experiments, Avery, MacLeod and McCarty finally concluded that DNA was the transforming principle and the genetic material. Scientists were still not convinced with their result, and they still continued believing on the theory that protein was the genetic material. It wasn't until 1952 when two other American scientists named Alfred Hershey and Martha-Chase performed with the definitive experiments to prove that DNA was indeed the genetic material. They worked on bacteriophages, which are viruses that infects or literally eats bacteria. During the time, bacteriophages were widely used as experimental models because these viruses were very simple and they showed rapid reproduction. So such simple bacteriophages consisted of only two components. They had a DNA core which was surrounded by a protein coat which is called as capsid. Let me briefly explain the mode of action of bacteriophages or how these viruses infect bacteria. And these facts were already known to the scientists during the time. As you see in the illustration, the first step is viral attachment. So viruses are going to be attaching to the receptors on the bacterial cell surface, and this would be followed by injection or penetration of the viral genetic material into the bacterial cell. The subsequent event would result in the multiplication of new viruses in the bacteria. And this happens because viral genome takes over or it hijacks the bacterial genome, and such bacterial cells are now going to be synthesizing more viral DNA and viral protein. And subsequently, this would cause the bacterial cell to undergo lysis, which is to say the bacterial cell is now going to be rupturing, and as a result of which they are going to be releasing all the newly synthesized viruses, which then would be infecting other bacterial cells. The question that remained to be confirmed by the scientists was, what was the molecular nature of this genetic material which was injected by the virus into the bacterial cell? Is this genetic material the protein of the bacteriophage, or is this genetic material DNA of the bacteriophage? To figure out which of the two components, protein or DNA of the virus, got injected into the bacteria, Hershey and Chase used two radioactive isotopes to label bacteriophages. And these two isotopes were S35 and P32. Now, why did they choose sulfur 35 and phosphorus 32? The reason is because sulfur is only found in the protein and not in the DNA, and phosphorus is found in the DNA and not in the protein. So these two isotopes can separately tag the protein and DNA of bacteriophages. Hershey and Chase designed two simple yet clever experiments in order to discover the genetic material. In the first experiment, they developed sulfur-35 labeled bacteriophages. Now, how they created these bacteriophages was by culturing bacteriophages in a medium containing sulfur-35. And because of which, all of these bacteriophages are going to be having their protein coat labeled with sulfur-35. In the second experiment, they developed phosphorus-32 labeled bacteriophages, which was again created by culturing the bacteriophages in a medium containing phosphorus-32. And as a result of which, all of these bacteriophages are going to be having their DNA labeled with phosphorus-32. Through these two clever methods, Hershey and Chase could now easily trace S35 tagged protein and P32 tagged DNA of bacteriophages in the bacterial cell. This is an outline of Hershey and Chase's experiments, and as I said earlier, they ran two separate experiments. In the first experiment, they created batches of bacteriophages in which their protein codes were labeled with sulfur-35. And in the second experiment, they created bacteriophages in which their DNA was labeled with phosphorus-32. And this step is known as radioactive labeling. The second step is called as infection, during which the viruses are allowed to separately infect on bacteria, and in this case, the bacterium is E. coli. Now, during the process of infection, viruses are going to be attaching on the surface of the bacterial cell, and they are then going to be injecting their so-called genetic material into the bacteria. After allowing enough time for the viruses to inject their genetic material, Hershey and Chase then separated the viral component from the bacterial cell, or they dislodged the viral particles from the bacterial cell by using a simple blender, and this step is known as blending. After separation, the next step is centrifugation, in which the suspension containing bacterial cells and viral particles were spun in the centrifuge machine, and they were thus separated based on their density. In this case, since bacterial cells are heavier than the viral particles, so all of the bacterial cells settled or sank to the bottom and formed the palate, whereas the lighter viral particles got suspended in the upper supernatant. And the last final step was the detection of radioactivity. In the first experiment, they used sulfur-35 to label the proteins of bacteriophages. Now, when they detected sulfur-35 in the palate and supernatant, what they found was sulfur-35 was predominantly present in the supernatant than in the palate. Palate had no radioactivity. In the second experiment, because they have used phosphorus-32 to label the DNA of the bacteriophages, when they detected phosphorus-32 in the palate and supernatant, what they noticed was phosphorus-32 was predominantly present in the palate than in the supernatant. Based on their observation that sulfur-35 was not detected in the palate or bacterial cell in the first experiment, they inferred that protein of the virus or protein of the phage did not get injected into the bacteria. On the other hand, from the second experimental result, detection of P32, phosphorus-32 in the palate or in the bacterial cell made them to infer that DNA of the virus entered into or got injected into the bacterial cell. To summarize the results of Hershey and Chase's experiments, the first result was sulfur-35 radioactivity was detected in the supernatant or viral particles and not in the palate or bacterial cells. And in the second result, phosphorus-32 radioactivity was detected in the palate or bacterial cells and not in the supernatant or viral particles. And based on these two results, Hershey and Chase finally concluded that it was the DNA which was the genetic material that got transferred or injected from the bacteriophage into the bacteria and not the protein. These definitive experiments conducted by Hershey and Chase finally confirmed DNA as the genetic material. So with this, I come to the end of today's video lesson on the search for the genetic material. I'll catch up with my next video lesson on the structure of DNA.